Welcome to this live stream. I'm delighted tonight to be with Dr. Richard Sternberg, someone who I've been following for many, many years, and I'm sure many of you watching are familiar with his work. Welcome to the podcast.
Thank you for having me.
So, for those of you who are aware of some of the credentials of our esteemed guest today, Dr. Sternberg is an evolutionary biologist with interests in the relation between genes and morphological homologies and the nature of genomic information. He holds 2 PhDs, one in biology, molecular evolution from Florida International University, and another in systems biology, theoretical biology from Bingham, I don't know how to pronounce that, Binghamton, it sounded like Birmingham University. From 2001-2007, he served as a staff scientist at the National Center for Biotechnology Information, and from 2001 to 2007 was a research associate at the Smithsonian National Museum of Natural History. Dr. Sternberg is presently a research scientist at the Biologic Institute, supported by a research fellowship from the Center for Science and Culture at the Discovery Institute. He is also a research collaborator at the National Museum of Natural History. Thank you so much for joining me tonight. So, Dr. Sternberg, today, more than ever, there's a lot of news about freedom of speech in certain places in the world where there's curtailment of freedom of speech. And your time in academia is an example of where free speech has been affected by dogma. So would you like to recall with us tonight in terms of how your time at the Smithsonian Museum and that episode with Maya and what it led to, and perhaps some lessons for people watching today?
Sure. So to begin with, I had questions about neo-Darwinism, really going back to the late 1980s throughout the 1990s. But I was neither a creationist nor an intelligent design supporter. However, within a year or so of being at the Smithsonian, I became intrigued by intelligent design. I would say I was somewhere between being lukewarm to friendly to it, but I was not a supporter of it. In 2003, 2004, I was contacted by Stephen Meyer. He had an article on the origins of the major taxonomic groups, mainly animal groups. And his question was whether or not this would be something that would be of more general interest. And as a bit of a backdrop, during that time, within the context of the museum, there was discussion about Michael Behe's Darwin's Black Box. There was discussion of ID. The book by Jonathan Wells, Icons of Evolution, had been published shortly before. I knew some curators, they had copies of it on their bookshelf, and so it was discussed. And I put feelers out, and the opinion that was given me was that yes, it's of interest, and should I consider Meyer's paper for publication, there probably would not be big to do about anything. So I decided to give it a go, see what happened. What I would say to your audience out there is that the word to keep in the back of your mind is naivete, because I had no clue what the hell I was getting myself into, really. And on top of that, I would also add presumption. I had presumed that I had some kind of security within the framework, and I had no idea that I certainly did not. So long story short is that the manuscript was sent out for peer review. There was a lot of back and forth, and based on the reviews that I got, based on a careful reading of the manuscript and having discussed it with colleagues, I came to the conclusion that Okay, as the editor of a rather well-established journal, one had been around since the late 1800s. One actually started with the discussion of Darwinism in general. I thought, well, this is going to be a very timely issue. And manuscript appeared, had one line in it. It's a long paper. Had one line in it saying that looking at all the various contenders, standard neo-Darwinism, self-organization theory, and you just go on down the list, there is no reason before the fact, a priori, to dismiss intelligent design as an alternative hypothesis. And that was, that one sentence is what caused problems. So to make a long story, a rather long and painful story short, I would say that there was such a reaction from outside groups. It wasn't really inside the museum initially, but there was such a strong reaction from various groups. There were people were making phone calls, they were sending emails, and I had to wade through a lot of false accusations, like I had received money to publish the paper, that I was actually not a scientist. One person said they thought I was a priest. Actually, I had disguised myself all this time as being a biologist, and just on down the list. So there was no peer review. I was all the peer reviewers, and so on. Or I actually wrote the article, and Stephen Meyer slapped his name on it. And things went downhill from there to the point that it was no longer. I just felt no longer safe actually going there. I had been told that if anything went missing, anything was stolen, anything that happened at all, I was the one who was going to be blamed for it. That I had the community had lost all trust in me because I had failed as a gatekeeper. And so what I did is I compiled all that had happened. I filed a whistleblower complaint with the Office of Special Counsel, and to just have them investigate. And they did investigate, but then what happened was a local federal judge ruled in DC that I lacked civil rights. And so I received a very lengthy report about the investigation they had conducted. Clearly, they were saying, the evidence is supporting your claims here, but we're now hamstrung by this judge who's saying effectively, it was myself and one other guy, that we lack the civil rights that's accorded to other individuals. And anyway, so that's it. It's something I don't want to ruminate on. Sure. Like I said, it was it shaved a few years off my life, I'm sure.
Yeah. And I think for a lot of people following the discussions between ID and neo-Darwinism, this was a watershed moment, just to show underneath the veneer, underneath the decorum, there is a lot of dogma. There is a lot of intolerance towards other perspectives. Now, to speak about today's topic and to sort of bring it in, I wanted your thoughts on a very famous interaction that happened here in the UK between the chief rabbi Jonathan Sacks, who himself was a philosopher, and Richard Dawkins. Many years ago, they actually had a debate. And during this debate, many things were discussed, but one of the things that the rabbi brought up was junk DNA. And he said, well, we used to think the genome was largely junk. We've now discovered it's not. And Dawkins replied, well, creationists are using this argument now, but of course natural selection wouldn't leave the genome stuck with a whole bunch of junk, and it would be efficient and it would sort of clear up the mess. So there's a lot of confusion online about junk DNA. You have people like Richard Dawkins who accept now that actually it's not really junk. Then you have people who are PZ Myers and others who are like, no, it's still junk. And then even some contemporary younger biologists like Daniel Stern, who Casey Luskin debated, again arguing against it. So let's first break it down one step at a time, if you don't mind, Dr. Sternberg. The very first thing is junk, just the term junk. What is the origin of this? And what evidence was there to actually categorize the genome as junk in the first place?
Well, the one, to my knowledge, who first called it a junk was Susumu Ono. In 1972, it was a paper titled "Why So Much Junk in Our Genomes," and there he was ruminating on duplicated genes and that, but also what has been called the C-value paradox. And so by the 60s, certainly by the early 70s, it was known that in the case of nucleated organisms, eukaryotes, the quantity of nuclear DNA can vary by 40,000 fold. You have some organisms that, like an amoeba, that can have very large genome sizes. You can have others, it can be like an orchid species that has a very small genome size. So no one was able to correlate the quantity of nuclear DNA with the complexity, the perceived complexity of an organism. So the idea of junk DNA that Ono proposed was rather timely. But it was really, I think, a combination. You go from 72 to 1980, you had several things occurred. One was, of course, Dawkins published his famous book, The Selfish Gene, 1976, first edition, where he makes the argument that what's really important is you have this conflict going on amongst these selfish replicators. The name of their game is simply to produce as many copies of themselves as possible. And then that was married to the idea, selfish DNA was married to the idea of junk DNA, really in 1980 with two papers that came out in Nature, one by Doolittle, W4 Doolittle and Carmen Sapienza, and the other by Leslie Orville and Francis Crick. And so to have such high-profile individuals giving the imprimatur and nihil obstat to these ideas was very influential. And nevertheless, there was ample evidence in the literature dating from the 30s on, and well-studied organisms like Drosophila to show that a lot of it simply is not junk DNA. And in fact, you remember around the time, so 1983, 3 years after the selfish DNA and junk DNA concepts had come to the fore, Barbara McClintock had won a Nobel Prize, and she won a Nobel Prize for studying what? Jumping genes. She had discovered them, and for the idea that a lot of the so-called junk DNA actually has a developmental role. So that, in a nutshell, is where the notion of junk came in.
Yeah. Thank you for that brilliant introduction into the term. I also wanted to get your thoughts, Dr. Sternberg, on the weaponization of junk DNA against believers, against people who believe in some form of design. I remember the British philosopher, and in particular a philosopher of science or philosopher of biology, Philip Kitcher, saying most of the genome is junk, so God needs to go back to school. Now, there's two things about this statement that I found quite startling. The first was that, as a philosopher, he should know better because of the problem of induction and underdetermination and all of these philosophical issues that science can change. So you shouldn't be categorical. And secondly, well, we're moving off from biology to theology here if we're going to talk about God and God going back to school if the genome is junk. But it seems like this was a knockdown argument that the neo-Darwinists or the atheist materialists actually had against design, right?
Yes, it was certainly, it was grist for their mill. I will say that I've known a number of good card-carrying Darwinians who never bought into the notion of junk DNA. So, so the field, I can't speak about today, but the field back in the 80s, 90s, 2000s To be a Darwinian did not necessarily mean that you bought into the notion that 90 some odd percent of our nuclear DNA is just simply garbage. No one bought into that necessarily. Those who were high profile proponents of it, and you say yes, some of them philosophers and should know better. It was several ideas had come up. One is that DNA became enshrined as being the book of life or the language of God, as Francis Collins referred to it as being the blueprint of the cell. And this had really been gaining momentum since the 1950s. So much so that I'm sure you've heard it. People say, "Oh, it's in my DNA," or it's just they use that term all the time. So if you have this idea that the essence or the core of one's self or one's dog or one's begonia, it doesn't matter. The core of it is DNA and DNA only. And then you add to that this core, this essence, this blueprint is 95% junkety junk. It just is nonsense. And what you've done is you've really, you've made a compelling case or seemingly compelling case for non-design because it's like if indeed DNA is the language of God, if indeed DNA is a blueprint, then why, when we look at it, do we see miles and miles and miles of just plain gibberish? No good designing entity, however conceived, so the argument goes, would put forth something like that. And this argument came around at the time when, unfortunately, and I want to underline, unfortunately, you had creationists here in the United States who were trying to get a version of biblical literalism passed as science to be taught in schools. And so this, if you will, augmented the design antibodies on the part of many Darwinians. And so they started looking for arguments that would, you could easily refute your corner creationists, and junk DNA fit the bill. Seemed to fit the bill.
Yeah. And just to add, that this particular strand of creationists were going with their reading of the Bible. I think that's also important because creationism is a very broad idea.
I'll say when I use the term, I'm referring to, and I'm not using necessarily pejoratively. I just usually in the 80s and I guess in the 90s, it was those who held to a young Earth. The Earth is 6,000, 10,000 years old. The whole shebang as far as it goes.
Yeah, and as I regularly point out when this is raised to me, that is their reading of the Bible. That's not necessarily what the Bible says. Okay. Additionally, I wanted to add, there were people, like you said, who weren't intelligent design proponents, but they held to the idea that junk, the genome is not junk. And then you had obviously people who were critics of neo-Darwinism, but they also held on to the idea that it's not primarily junk. So you pointed out McClintock and her work, and now James Shapiro, her successor, he was adamant in the 90s that actually this is not going to be found to be junk. And I believe you were also of that opinion that, we will move away from this. Now, I remember, I believe it was some interaction between yourself and Shapiro. I've forgotten where it was. I think Casey Luskin alluded to that. So some sort of interaction, whether it was a paper in which you were referenced by Shapiro or whatever it was. But could you go into a little bit of that history and why you guys held on to this, despite the majority view being that it is still junk?
Yes. So what happened in my case, I'll get into my interactions with Jim in a moment, but in the 80s and certainly in the 90s, I was very impressed by McClintock's Nobel Prize address. So that was published 1984 in Science. And she presented this beautiful holistic model of what a genome in effect has to be. And it was clearly written by a woman of magnificent intelligence, but one who was really able to peer into a cell. Her mindset, I think, is best captured by a book by Evelyn Fox Keller titled "A Feeling for the Organism." So she was she tied everything in. She kept cells in the front view as well as the organism, and then she married that with genetics. I became very impressed with that. The problem was my professors and those that I knew at the time, and I had kind of moved over from philosophy. I initially wanted to be a philosopher into genetics, primarily because of reading that paper. I was enthralled by it, and they wanted to cool my ardor. And the way to do that is they would ply me with, "Oh, you have to read this paper about it was junk." And so what I decided to do, which I would not recommend for most graduate students, is I spent an enormous amount of time in the library going through journals like Chromosoma, Journal of Cell Biology, just trying to find empirical evidence for what these junk regions were doing, being told all the while that it was a fool's errand. I was going to come up empty-handed. And really, by the end of the 80s, I had file boxes full of papers published in the 30s, 40s, 50s on that gave, in my opinion, good experimental evidence that no, a lot of these regions are not junk. So, for example, one of the ideas sold was that, you can, the reason junk DNA can be so selfish is that there's no cost to the organism for having extra copies of it. So you can just make as many extra copies. And so, cellular currency is no object. So redundancy can rule the day. And what I found was that no, actually in the 50s, they did experiments with fruit flies where the Y chromosome was at that time considered largely inert. It's largely repetitive DNA, just miles and miles and miles of repeats. And what they found, though, is that if you doubled the dosage or tripled the dosage, it had adverse effects on embryogenesis. And there were other studies like that. So they showed that, no, actually, there is a cost to having, large amounts of excess DNA. And I just would find example after example. So now fast forwarding to the early 1990s, I met, I'd always been an admirer of Jim. I got to meet him in the flesh in Athens, Georgia at a conference. And he wrote a paper, it was a very short paper. One of it was Genomes as Smart Systems, where he was looking at it from, I would consider, a very sophisticated cybernetical view of what a genome has to be. And then he also looked at what we knew about jumping genes and other families of repetitive DNA sequences, and concluded that at the very least they're engaged in some form of what he called natural genetic engineering. So yeah, genetic engineering had really come to the fore in the late 70s to mid 80s. And he had provided really good research using E. coli primarily to show that, yes, you have some developmentally programmed natural genomic engineering taking place. And so I maintained correspondence with Jim. And then in the mid 2000s, so this was, at that time, I was working as a staff scientist with the National Center for Biotechnology Information, which is part of the National Institutes of Health. And so that was my bread and butter. And what I had noticed certainly was that in 2000, when you had two groups had sequenced the quote unquote human genome. And the idea was, just by wading through these miles and miles of A's, C's, G's, and T's, one would be able to compute the organism. That was the idea. And then within a few short years, we're going to have designer drugs based on someone's genetic profile. Okay. The problem was there was all this data and no one knew how to decipher it. And so I had taken a step back and I'm saying, well, we actually know a lot of things from cell biology, from chromosome biology. And Jim approached me about co-authoring 2 papers on the subject. And so for about a year and a half, what I would do is I would just amass the data that we had, relying on Jim's intellect. He was the sailor piloting that ship, and we came up with, I think, a very prescient notion of what exactly so-called junk DNA is doing. So much so that I concluded independently of Jim, but I just decided to put it out in a paper that I think that the majority of DNA sequences in a particular organism probably are functional in some way. And I've only strengthened my my view at that time. But Jim, I would say, really by the late 70s or by the 80s, if ever. Let me rephrase that. Certainly by that time, and I'm not sure he ever adhered to any notion of junk DNA, but his conclusion was that we simply lacked the conceptual framework to properly interpret the data, and I became convinced of that, and really by, I would say, 1992, 1993. The only way one could maintain the notion of junk DNA is if one stayed within the framework of just simply evolutionary genetics, where you're dealing with essentially mathematical manipulation of symbolisms, and it's a little bit divorced from the organism itself. But if you started focusing on cells, on the organism, I think one would be very hard pressed to have stayed with the junk DNA notion.
Absolutely. And I really
Sorry
like
for
that.
rambling.
No. This is gold for me. I'm really, I'm sure the audience is enjoying this as well. So that was a really interesting period in the 90s. And I think one of the things that stands out as well is that, and this is when I was doing I did my master's thesis on junk DNA and methodological adaptationism. So I looked into the history of these things. And one of the things I found interesting was that is almost self-evident at the time for evolutionary biologists that anything that doesn't code is junk. That was just a given. It wasn't even questioned. Maybe non-coding has a function. Another thing which I feel, and I want your thoughts on this, was because the air in evolutionary sciences was dominated by reductionism, everything had to be causal from a gene perspective.
Mm-hmm.
The idea that, as McClintock thought, the genome is an organ, a highly sensitive organ of the cell, that stopped people thinking out of the box. Now, you mentioned the conceptual framework. The conceptual framework, in a way, correct me if I'm wrong, it could be the conceptual framework was the integrative, holistic understanding of the unity of the organism and how all the parts work together, as opposed to the gene-centric one-way causation methodology that most of biology is dominated by.
Mm-hmm. Well, I think that's correct. I think there are two concepts I don't like, and that's not because I think they're bankrupt. I think they did a lot of damage. One is the sequence hypothesis. I think it should be called the sequence thesis. Stronger, a little bit, in my opinion, stronger than a hypothesis of Francis Crick, which is based on the notion that proteins are the most important part of a cell, and therefore the most important thing DNA can be doing is coding for proteins. Automatically, if something does not code for protein, it got put into that side category of probably not being that important. And then, of course, the central dogma, also given by Crick, which was the idea that you have this linear flow of information, DNA, RNA to protein. And yes, RNA can be reverse transcribed back into DNA. Again, it's not that it's incorrect, but what it did is it was a Procrustean bed. So if you had any data that simply did not fit into the dimensions of that theoretical bed, those parts got lopped off. And so that was, I think that combined with the selfish DNA or the selfish gene notion, those were the things that really firmed up the reductionism of the 1980s, 1990s. I had many professors who believed that it was all so simple. Effectively, we know everything there is to know about the organism. An E. coli is a beetle, is an elephant. It's all just these interchangeable parts. I remember Emil Zuckerkandl wrote that if one took the genes, just the protein coding regions, and some control elements, certainly of a particular higher organism, then by rearranging them, one could create almost any other higher organism. So that was the thinking. And any depictions of order, the name of the game was to explain it by there being some underlying disorder that was giving the illusion of organization.
Yeah. Fascinating times and really interesting from a historical perspective, because we can see the convergence of many different lines of inquiry which were pushing against that narrative. So now we go to the early 2000s. We have the Nature paper of the human genome sequence of 2001, which Shapiro and Noble and others have used to show genome reorganization. So this involves yeast, worms, flies, the humans and other vertebrates, and showing that large parts of the genome was actually reorganized. It wasn't point mutations accumulated by natural selection. So what was the significance of that paper at the time?
Now, which paper are you referring to?
This is the 2001 human sequencing, the first time that it was sequenced, and it was published in Nature.
Yeah, I'm thinking, well, I think the significance of it was, for me, because I had just, I'd started at the NIH in around that time. But the significance was that there are so few protein coding genes in the human genome.
Yeah.
So the idea was that humans had, they speculated roughly 100,000 protein coding genes. A fly would have maybe 20 some odd thousand. And one of the discoveries was that actually, when you look at a nematode, a small worm-like organism, or a fruit fly, or a human being, or a cactus, they actually have pretty much the same protein coding regions. And the differences that seemed to make a difference were lying outside of that. So that was one thing. The other thing that was, I think, startling was that in our case, over 50%, and maybe over 60%, consists of several families of jumping genes, which resemble very much retroviral-like elements, or they have pretty much the same complement in general. And so the pervasiveness of non-protein coding DNA, I think, was stunning for many. They expected humans to have far, far more genes. And then also to explain some of the differentiation that occurs in, say, the immune system, this has been known really since the 70s, is, of course, you have a number of somatic DNA rearrangements that take place. You have a number of mechanisms that diversify DNA as development is going on. And we also had indications around that time that this appears to be taking place during central nervous system development. And now we know in humans and in mice, et cetera, yes, indeed, you do have a lot of DNA that's being swapped around as neurons are developing. So these were surprises, but what prompted, for example, the ENCODE project was that on the one hand, you could, in a laboratory, you could identify that, yeah, there are 100 proteins or 100,000 protein isoforms and more than that, but there were a limited set of protein coding regions. So there had to be a mechanism for somehow explaining how to, going from that limited subset to a much broader set. There's something going on here, and who's in the driver's seat? That was the question of what's going on. And then shortly thereafter, this is 2007, 2008, then it became clear that most of the activity that's going on in chromosomes in any given cell lineage, in any given cell, at least in human beings and mice, a few others, is taking place in the non-protein coding regions, which was contrary to all expectations.
It was, and it certainly put to bed this idea that you have a gene for this, a gene for a particular function. And, this belief that was hyped up by the likes of Francis Collins and others, that we will discover the secret to life. We will discover, almost to the level, obviously he didn't put it in these words, these are just the sort of colorful ways we can remember the idea. You could walk around and give your doctor, your genome, and they'll be able to tell you, well, you're going to get cancer, you're going to get heart disease, you're going to get this, you're going to get that. The association studies showed that the association between the genes and the particular effects later on, there was no clear correlation. In fact, I remember in the debate between Richard Dawkins and Dennis Noble, one of the central points that Noble was pointing out is the association studies have fundamentally failed to show a correlation.
Mm-hmm. Yeah, that's interesting. Well, around the mid 2000s, early to mid 2000s, a project was started to sequence the genomes of various dog breeds. So there, you can name the breed, you can name the temperament. And by comparing, say, the DNA sequence of a Labrador Retriever with that of a Dachshund, with that of a Rottweiler, et cetera, then one would be able to locate where these particular temperamental or a gene for aggressiveness, say, or a gene for a docile temperament, say, where these would be located. One did not have to spend a lot of time in the literature to realize, it was much harder than that. The problems are compounded. So looking from that period, early to mid 2000s to the day, the problem is compounded where in many model organisms where you have very good genetic studies, and I'm thinking of Baker's yeast, Saccharomyces cerevisiae, which I consider a premier example. You run, even there, when you have these genome-wide association studies, you run into a problem that's called missing heritability, where there appears, yes, there are things that are inherited, but when you try to find the underlying DNA sequences responsible to it, it becomes problematic. Another thing that has become clear, so second thing I'll mention on this of three, is that, this has been found in yeast, is that some genes which behave in a good Mendelian manner, of the kind you would have learned in school, where you can do your Punnett square, depending on their background, you can take these nice Mendelian genes and same species, Baker's yeast, and you put them on a different genetic background, and suddenly they don't behave in a Mendelian manner anymore. They start behaving in more of a what's called a polygenic or a quantitative manner. So you have that. And the third thing is this looking at traits, say human height. So it would be the, which is of course variable, highly variable. and there appear to be, when they've mapped them, there appears to be about 10,000 regions along the chromosomes that can be correlated with differences in shortness and tallness and what have you. But when you look at how they're expressed, the model that has come up is they appear to be what's called omnigenic, meaning it appears to be everything's involved in somehow influencing the particular trait that's come along. It's not one particular part of DNA on a particular chromosome. It appears to be all over the place. Everything is being factored in. So the nice clean models of genetics and evolutionary change that you were undoubtedly taught, I was undoubtedly taught, I think they have come under considerable scrutiny, and many people are looking around for alternatives.
Yeah,
as we speak.
Brilliant. And I'm glad you brought the omnigenic perspective, because that is really key to understanding, again, how a conceptual paradigm shift would lead you to reading the data in a totally different way. And I think the way that Noble explains that is, it's like an organ, the old organs with all the different parts, and you can think of those as genes, and the combinations of the music that can be generated from the same set is almost infinite, right?
Yeah, it is. Yeah, it would be infinite, actually. Yeah.
And,
but then that raises the question of who's playing the organ.
yeah. And also, what's interesting about this is, if we look at reductive biology versus integrative biology today as it stands, we find that the same sort of barriers that you were facing in terms of expressing your views when you were at the Smithsonian Museum, and the sort of challenges that you had there, well, we're having the same sort of challenges at a broader level when it comes to the discussions between holism and reductive biology. We are finding that the likes of third way evolutionists, and even, you could say, extended evolutionary synthesis proponents, those discussions are being shut down or rubbished, or being downplayed. And you find a lot of people pushing back and bringing that archaic understanding of biology and still trying to make it work. It's almost like, getting everybody to give up their mobile phones and taking us back to landlines.
Oh, yeah. I think it certainly is that way. I mean, Thomas Hunt Morgan, Edmund Beecher Wilson. So these figures from the early part of the 20th century. Morgan, of course, famously goes from embryology, sets up the fly room, but he stated from the outset that any relation between a part of a chromosome and what we call the phenotype of an organism is going to be one to many, and it's going to be many to one. So you're probably not going to be able to compute things out that way. Edmund Beecher Wilson, he had this very integrative view. He thought, yes, chromosomes are essential for heredity, but so is the cytoplasm. So is everything else that's going on there. And Morgan, in, I think it's a paper from 1909, 1910, I was reading it a few weeks back, he stated that the reductionist way is the easiest way to go. It's easy because you can grind things up, you can do analyses, you can crank out papers, you seem to be able to make a lot of progress. You can get some traction. But he said it's very limited into where it will take you. He said the holistic way, you look at it from afar and it seems rather intangible, a bit muddled, needs to be clarified. That's true, but what it does, for all of its inadequacies or maybe initial limitations, I'd phrase it that way, nevertheless will lead one into territories that they had no clue existed. So it tends to be more productive in the end. And I think that the thinking of someone like a Dennis Noble, of a James Shapiro, and many many others, of a Michael Levin, for example, um, there it's a holistic picture, but it's one that does not hinge on nebulous vital forces. It doesn't hinge on, I mean, it's something that you can model. It's something that you can explore from a number of angles, and it's not closing doors; it's opening
Yeah.
doors actually. So it's more productive in that sense.
Absolutely, absolutely. And taking, as Sidney Brenner has argued, the level of abstraction to the cell as opposed to the genome, it opens up those epistemic doors, right?
Yeah,
And I think this is a good time to bring in the idea that a lot more is inherited than the DNA. So
yeah,
the entire cell structure is self-templating. You don't even have the genome doesn't even have the coding for the membrane of the cell, and the conditional logic of life, the if then this, and all the other conditional logical things, you will find within the membrane, and that will actually activate parts of the genome. So you've got all this conditional logic there. So that in of itself is a brute fact against the idea that the DNA, the genome, is the blueprint of life, because not all the conditional logic is there, and it's inert until, like James Shapiro calls it, it's a read rewrite file.
yeah,
It has to be, it has to be activated by something else.
yeah. Yeah, I mean, I've always been amazed by if anyone has isolated DNA, and you see it in its various forms. I mean, you can purchase from, let's say, like Sigma Chemical, a vial of herring sperm DNA, and you look at it. It's in the crystalline form, but it's completely inert. It will do absolutely nothing, and it's about as it's like any other polymer. It's a highly stable chemical polymer. That's what it is. It does not self-reproduce. It does not self-transcribe. But looking at other things that are inherited, um, one thing, this idea that it was chromosomes alone, we knew that really by the latter part of the 1800s. It was known that the male sperm, at least in many animals, including humans, contributes a centrosome. So eggs do not have a centrosome. It's the male sperm that brings this non-chromosomal body, the centrosome, into the ovum, which then divides and provides the apparatus for all future cell division. It's a self-propagating system. So you have that example. Some other examples I've seen recently is where they take cell-free extracts. And these are with frog eggs. So they're large. You can get a lot of the materials, but when you remove the nucleus, so the question is, how much of development can you get from an egg and that you've thrown the nucleus out? You just want to see how far it will get. Well, you'd be surprised how far it can get. So it means a lot of the information, as we call it, is there in the cytoplasm, and it will perpetuate itself. It will eventually peter out. It will die. But the fact is that in the absence of DNA, the same is true in fruit fly. You can get considerable amount of development before you need to start reading and writing DNA codes.
Absolutely, absolutely. And as we're coming to the end of our session, I want to ask you, I guess it's a good time to bring in, there's a lot of discussion now in the literature about physiological functional networks and how they arbitrate between the genotype and the phenotype, the idea of genetic buffering, and how junk DNA plays a role within that broader discussion. What are some of your thoughts on this?
Well, I think it's true. The many of the sequences that are called junk are known to have roles in chromosome organization the following ways. One is that they bind proteins and ribonucleoproteins that act as scaffolding. They basically attach regions of the chromosome to the nuclear envelope, or they're contacted by cytoskeleton fibers, tethering agents that even can go from the cell exterior all the way into the nucleus. So they appear to be part of some kind of mechanical transduction system that's going on there. You can change the shape of a cell. You can force cells to adopt a particular geometry. And it's been known since the 90s that once you do that, you change gene expression.
Yeah.
Now, you haven't altered anything chemically necessarily, but you have changed what is called the tensegrity of the system. You forced it to adopt a certain geometry, and so you have like a causally downward flow of data that says, hey, we need to switch these genes on, we need to turn these genes off. And in many cases, the elements that are the part of that transduction system are the non-protein coding elements.
Yes.
You know,
Yes.
and we're finding some fantastic examples. One of my favorite examples of this concerns nocturnal mammals. The photoreceptor rod cells, the nuclei have a particular morphology. It's easily discernible from other cell types, and what it is the nucleus as a whole. So in toto, the nucleus becomes an optical device. It scavenges photons, and so the way sequences are arrayed in the nucleus are done so to optimize nocturnal vision. So here's a case where effectively, a lot of the DNA is playing a role, albeit it's not a protein coding role. Another case is in certain white blood cells that we have, and this is true in shrimp, this is true in humans. When those cells, their role is effectively to die in the presence of a virus or bacteria, the nucleus then is lysed or opened up, and the DNA spools out. It's like a bowl of spaghetti that goes in cooked spaghetti, it goes in all directions, and it traps the viruses and the bacteria there. So there the DNA is playing a filtration role, but all of it is. And there are other examples where, in particular cell types, where it looks like every single DNA molecule is playing a role in the context of a greater organizational system. And those are just three. I could go on.
Absolutely.
For hours.
Yeah, absolutely. And thank you for that fascinating insight into the world of the unseen. I wanted to add one more thing. As you were speaking, I remembered how this fits in broadly with the idea of physiology guiding evolution. Those epigenetic markings, if the environment remains the same and they are repeatedly invoked, they do become assimilated within the genome. So you get real genetic change. Yeah. I mean, I believe you referred earlier to the work in the 50s. So Conrad Waddington obviously worked on this. But but today, I mean, I think I was just a few days ago, I was reading something that was just published, I think back in March. I mean, we're finding lots and lots of evidence of this. And what's really interesting is not only does this give us a wider appreciation of, again, McClintock's idea of the genome being a highly sensitive organ of the cell, it also goes to highlight that there's a lot of areas in which we haven't even began to find out what processes are actually at play. There's a whole array of areas which we haven't even started to push the spotlight in that area. And that's one of the things which Shapiro pointed out in a podcast I did with him just a few months ago that there are processes that we cannot even imagine currently.
Oh, I'm convinced of it. I mean, when you look at the just how do you go from a single unicellular organism that I were once, we were a zygote, we were just a single cell, and now we consist of trillions of cells, all differentiated into. Now the estimate I think is we have well over 1,000 cell types, and then but we have it's not just 20 or 30 or 100 as we used to think. But they able to show that you have all these different cell types, you have cell subtypes organized into tissues, etc. How in the world does this happen? And we know, and this was something that Jim had shown in the 70s and 80s, that with just bacterial colonies, something that is seemingly as simple as that, they'll form these complicated flower-like patterns. So will yeast cells. They effectively become a multicellular organism. It's a bacterial colony. But then you start getting these intriguing patterns of crosstalk that's going on, and cooperation, and division of labor that's going on. If you look at the literature, we know very little about how that happens. And that we're talking about microorganisms. So when it comes to something like a human being or a whale or a rose bush, I mean, I think we are, I'm not sure we'll ever have it all figured out. I think it's hubris or tantamount to hubris to assume we'll have it all figured out. But I expect that unless something unfortunate should happen to free scientific inquiry, I think 10 years from now, we'll be shocked at what we've learned. And 10 years after that, we'll be even more shocked at how ignorant we were. And so it's very exciting. It's also very humbling.
It is indeed. It is indeed. So on that delightful note, we'll conclude our session. Thank you so much, Dr. Sternberg, for your time. I really do appreciate your work. I've been following it for many, many years. And leave some links in the description for anyone that wants to follow up on your work. Any parting words you want to give us before we finish?
I'll just say that junk DNA is not. That's it.
That's the upshot. That's the upshot of our discussion. Thank you so much.
Thank you.
Cheers.